Okay, everybody, so we're down to our last topic of the term. And what we're going to do is we're going to continue on probabilities. But now what we're going to do is we're not going to do just autosomes, which is what we've been doing. Remember, there's 24 different chromosomes. From each parent, you get 23, right? 22 autosomes, which is 1 through 22, plus an X or a Y. And so there's 24 different chromosomes, 1 through 22, but then you have X and Y. And so what we're going to do is now we're going to bring those in. However, what we have to do is kind of go back and see how some of this was established. Okay, so let's take a look. So if we remember, the model that we saw under Mendel was pea plants, right? It was pea plants. In looking at sex linkage, and this wasn't too much later, maybe about 30 years later or so after Mendel published his stuff, a lot of geneticists were doing fly genetics, and that continues till this day. And so what we're looking at is we're looking at not the blowfly. The blowfly is that big housefly that you see all the time. What we're looking at is we're looking at Drosophila, okay, Drosophila melanogaster. This is a fruit fly, so it's tiny. It's not those big flies that come in when you leave the door open at home, okay? And so when we look at Drosophila, it has advantages over pea plants, okay? First of all, it has a short life cycle, about 2 weeks or less. We're talking about 12 days to actually get a full adult. And you have many more progenies than you would in pea plants. In pea plants, you could have hundreds of peas, whereas when you mate flies, you can get thousands of flies. And they're easy to grow. They're absolutely easy to grow. I remember I did my after I was done with graduate school, I did a 4-year fellowship at the City of Hope Cancer Center. And at the time, I don't think they have it anymore, but at the time they had a fly facility. And it was interesting because you would walk in to the facility and it kind of looked like a library, except there was no carpet, in that you had the shelves, right? The shelves where you usually have all the books. But what you would notice is on those shelves, instead, is you would have these milk bottles. Okay, the milk bottles look like those glass bottles that they sell at Starbucks when you buy one of those iced, frappuccinos, but they come in the bottles, right? So they look like that. And instead of having books on the shelves, they just had dozens and dozens and dozens of these bottles on each shelf. And you had these shelves that would go, again, just look like a library. And in that bottle, what you would see is you would just see hundreds, maybe dozens, hundreds, even thousands of flies. They were very easy to grow. It was interesting because, and I have a lot of respect for fly geneticists. The amount of work that they do is just so painstaking. What they would do is they would get 2 particular type of flies that they wanted to mate, right? And they would mate them, and it was in these bottles, and then they would hatch, right? Those are what we typically call the maggots, right? And growing them is very easy. When things first started, what you would do is you would just take mashed bananas and put them at the bottom of the bottle. And that was enough nutrients for just thousands and thousands of flies. And what was funny is you would walk into the fly facility and you'd notice you'd be swatting flies away from you, saying like, all these experiments are getting out. It wasn't like a swarm. It was just like every now and then you'd see one, you'd swat it away and think, okay, some of these are getting away. So I'm not sure how accurate the data was going to be. I'm sure they did a good job. But the counting process was really interesting because sometimes they were looking at genes that control development, in which case you would look at, well, what do the wings look like? What do the eyes look like? And so what would happen is you would have these bottles, filled with flies, right? And you're like, okay, they're mature enough now that we are going to count them. And so, but if you open up the bottle, then they all just fly away, right? They don't wait. And so the way that they would do it, I always found very interesting. And I know that in some of our curriculum, they do some fly labs, but I don't know if they do it this way. What they would do is they would take a sponge, just picture a sponge that's like, 6 inches by 8 inches. And on the side of the sponge, there would be this hole. And what you do is you take a hose and you stick it into the side of the sponge. And then the other end of the hose would be hooked up to a tank. And that it would look like almost like a tall helium tank, same type of tank, except it would have CO2 in it. Now, I don't know if you guys ever did this when you guys were young and you played around. Well, you would take a bag, like a brown paper bag, and you would put it over your mouth and your nose, and you would breathe and huff in and out, huff in and out. You would notice that you would get lightheaded. And I never did this, but I had friends who did this where they were huff and puff and they were trying to pass out. Okay. And so that effect isn't limited to humans. So if you start to hyperventilate into this bag, and you got all the CO2 building up and the oxygen is all gone, you're going to pass out. And all your friends are going to point at you while you're lying there on the ground laughing. So what's going to happen is you're going to take this sponge and you are going to, again, connect that hose. You're going to connect that hose to the CO2 tank. You're going to then gently turn on the valve. You're going to turn the valve and you're going to hear this hissing sound. The CO2 is starting to come out of all of those holes in the sponge. And what you're going to do is you're going to take your bottle filled with flies. You're going to put it right above your sponge, right, which is hissing now because it's got all the CO2 coming out of it. You are going to take the cap off, which is usually just, we're going to see it. It's not like a bottle cap. It's like a sponge plunger shoved into the top. We're going to see it. You take your bottle and quickly, right, you're gonna tip it over. You're gonna pull out the sponge stopper and you're gonna dump all those flies on the sponge and they all pass out. And then you point at them and then you laugh. And so as they are passed out, now what you can do is you can take a big magnifying glass and one by one, and this is why I have a lot of respect for fly geneticists, one by one, under the magnifying glass, it's a big magnifying glass, sometimes maybe 6 inches across, and it's on an arm, kind of the way you have vanity mirrors, you can kind of position them and so forth. Same thing, you have it like on this arm. And so you position it between you and the sponge. And now with tweezers, you can start counting all the flies. So if you're looking at eyes, right, red eyes versus white eyes, you start to count red, red. And you might have 2,000 flies to count. Or you might be looking at the body shape or the wings or whatever it is that you're looking at. And so fly people are just different people, lots of patience. And so it's, I remember just kind of fun to remember those times when City of Hope did have their fly facility. Now, when we think about the nomenclature, in other words, how we name the genes. So we've been doing big A, little a, right? That's the nomenclature. So I have a question. How do you keep track of the ones you've already counted? Yeah, what you want to do is you want to put them on one side of the sponge. And when you've counted them, you move them to the other side of the sponge, right? So you don't just dip them all in the middle. So the ones on the left side of the sponge are the ones you have to count. And then you look, you count it. Or I don't know, it may be that once they counted them, they didn't care about them, so you can dump them in alcohol, or I'm not sure exactly what they ever did with them. Once you count them, unless you wanted a few of them to mate for your next experiment, you don't really need them. And I don't think they would let them out. So somehow I think they discarded them. Okay. I don't know if the CO2 was toxic after a while, because after a while, if you don't have enough oxygen, at least with humans, right? We see this with drowning or any number of ways that you lack oxygen, a human dies. It may be the same for a fly. I'm not sure. I'm not sure. I didn't stick around long enough to see that part. And I had no intention of being a fly geneticist. So with regard to the name nomenclature, what we've been doing is big A, little a, big A, little a. And so what we're going to do is we're going to look at a new nomenclature that fly geneticists use. And at first, it seems like a big pain. It's like, why would they do it this way? But after a while, you actually get used to it and you don't even give it a second thought. But at the beginning, it just seems a little bit of a pain. So let's walk through this. Let's walk through this. With the nomenclature of fly geneticists, what they do is they keep track of a couple of things. First of all, they're going to have a symbol for the trait, just like we did with yellow and green peas, right? We had the Y, big Y yellow, small y green. So they're going to do the same thing. Okay, they're going to have different symbols, big, small. But they're also going to use a plus here. Let me show you what I'm talking about. Let's do an example. So, for example, if you are looking at flies and you're looking at eye color, okay, always the mutant is what is going to give you your symbol. So if you remember on the previous slide, flies, and remember we're talking about fruit flies, flies tend to have red eyes. That is their normal. But you can find mutants with white eyes. So the mutant always establishes your symbol. So let's say we're looking at eyes, red eyes versus white eyes. And so this is what we would do. We would write W, and that would mean that the fly has white eyes. Okay. Now, they do something that at first is a little confusing, but you'll see it'll go away pretty, in terms of the confusion, it'll go away. If it's normal, in other words, if it has red eyes, what they will do is they will put a plus on it. And we can think about it this way. If you think that being normal is a positive thing, you can use that play on words to remember that if it's normal, in this case, Drosophila usually has red eyes. You put a plus. So this would mean that the fly has white eyes. It's a W. If it's a W plus, it means it has normal eyes, in which case it's red. Now, what we're not going to do, but it still exists, is see, this is a small case W, which means the mutant is recessive. If we used a big W, that is also white. You know, let me get rid of this line here. Okay. If we use big W, that is also white. And then we would use capital W plus, that is also red. So why do we have two? Why do we have lowercase and uppercase? Because in this case, since the W, which stands for white, right, is lowercase, it means that the white is recessive. Over here, if it was big W, it would mean the white is dominant. Most of these mutations are recessive. So most of what we're going to do is lowercase. But that's what it means. So you're like, why are they doing this? It was so nice with just big A, little a. We're going to see it's actually going to help us. And pretty soon it's not going to be that complicated. So again, we're not doing big Y, little y the way we used to, right? In this case, we're using the same symbol, little w, but a plus is added if it's normal, Let's put this into play. Well, and just a little bit. Let me explain a couple of things first. So sex chromosomes, again, we really didn't understand a lot of inheritance. Remember, it took Mendel in the 1860s to figure out how this stuff happened. And that stuff was lost for about 30 years, okay? Sex chromosomes were discovered around the turn of the century, but they noticed that when the chromosomes were lining up, remember we have 46, there was a mismatched pair in some individuals, not female individuals, right? Because females would have XX, and so it didn't look mismatched. But in males, there was a mismatched pair. That's because they weren't the same size. So in males, what you would get is you would get this small chromosome paired up with this large chromosome. So we know that this is a Y and this is an X. And so they didn't really know the role that this played, and it really was throwing off some people who were trying to figure out how things were being inherited. Again, they hadn't rediscovered Mendel's stuff just yet. And so, the Y chromosome really doesn't have a lot of genes on them. Some people think that the genes on the X chromosome are somehow sex associated genes. That's really not the case. Remember, we all have X chromosomes. We all have X chromosomes, whether we're male or female. And so the genes on the X chromosome, they are part of our physiology that in many cases have nothing to do with sexual function or sexual features, organs, secondary organs. They are just regular aspects of physiology because we all have them. The Y chromosome is a little bit different though. The Y chromosome doesn't have very many genes. Right now it's estimated that it has somewhere around 20. Some people say 19, some people say 23. And those genes, the majority of them do have some function in like sperm development or other functions that have to do with hair growth, and usually not in the head. Usually there have been reports that, there are some genes that control hair growth on the ear, right? On the ear. And those are on the Y gene. So, but there aren't a whole lot of genes. Again, you're going to find about 20 or so, okay, 20. It was in 1910 that we saw a big jump in our understanding, and that was because of the fly room. Now, this was at Columbia University, and this is Dr. Thomas Morgan's laboratory. And the reason we saw a huge jump is because of the student that worked there. Now, in this lab, so he was an experimental biologist. He was trained in embryology. He was looking at development. But it was his student, Calvin Bridges, that actually made some of these discoveries. In particular, really what he did was he identified a mutant. Okay, so see, here are the glass bottles. Right? You can notice there are some bottles at Starbucks that look kind of like this. This is a sponge stopper. These can be mashed bananas. Now they sell synthetic food for the flies. It actually looks blue, so it looks like a sky blue almost down here. But you can still use bananas if you wanted to. So Calvin Bridges, he was working in the lab, and at the time he was, a dishwasher among other things. And as he was washing the bottles, he discovered a white-eyed fly. And they used that mutant then to do some crossing, right? You crossed it with other flies to actually look at the way the trait was inherited. And so it just so happens that eye color in flies is a gene on the Y chromosome. So, and it's interesting because then what would happen was you look at the inheritance patterns and they didn't make sense. And some people then still had a problem with some of Mendel's things, because once you start to do XY, it doesn't look like the autosomes, like the big A, little a that we've been talking about. Let's take a look. Okay, I think this is animated, right? Okay, let me just get the genotypes up. Okay, so here's one experiment that was done where you have a mating of flies. So here's a male fly. I'm sorry, this is a female fly, and we can tell because it has 2 copies of the gene, right? And over here we have a male fly. It has 1 copy plus the Y chromosome, right? And so this is red eyes, as you can see, and this is white eyes, okay? And when you cross that, what you would get is you would get half of your population female red, half your population male red. Now, at the start, that looks absolutely fine. Right? That looks absolutely fine. As a matter of fact, you can do a Punnett square and see how all these things actually do come out pretty well. In other words, this is what you would anticipate. You just say, okay, well, then the red is dominant. And we know that to be the case. Everything looked fine, except in the F2, something went wrong. When they crossed the F1, right, so you would get a red fly, which was female, a red-eyed fly, which is male, and you crossed it. And this was the ratio that they got. Can anybody tell me, you can unmute or you can put it in the chat, what is wrong with this outcome? Here's what you got. Here's what they observed. What is wrong with this outcome? Anybody see anything wrong? This is the F2 here. This is coming from so the Punnett squares still work, right? So if you do W plus, there's the female, and then you do the Punnett square, W plus Y, then everything works the same as before. W plus, W plus. Remember, the plus means normal, so this is red eye. W plus, and then the mutant is recessive, so W plus W, this is also red eye. This is W plus Y, that's a red eye. We got W, Y, that's white. That's where these come from. And here's what they got. Here are the phenotypes. Does anybody notice something weird when you look at this here? It may not pop out at you, but there is not a single white-eyed female. We have white-eyed males. We have red-eyed males. We have red-eyed females. There isn't a single white-eyed female. And look, you're talking about thousands. What is this? 3,000, more than 4,000 flies. More than 4,000 flies, and you're not getting white-eyed females? That doesn't make sense. Half of the females, well, depending on what the makeup of the parents are, but some percentage of them should be white, and they're not getting that. They're not getting that. What is it that's going on? And so if you were to do, so this is the second cross that they did. And so in this case, this fly, well, you guys should be able to tell me what this fly looks like. This was from the parent. Actually, this is one of the parents. So this fly, because it's a male, it's going to have a Y. It has white eyes. So this is a W. This female is from the F1, from the previous one. And so this one Actually, I think I have this animated. Oh, yeah, there it is. Okay, I should have checked that first. Animated these. Okay. And so this one is W plus W, right, from the F1. And they start to look at the progeny. And the thing about the progeny is they weren't lining up the way that they thought. Again, if you were to cross these, and they were autosomal, you would expect different ratios than they were getting when they were using X's and Y's. Let's look here. Let me see if I have any. No animation here. Okay. Okay, so you guys should be able to tell me what the genotypes of these flies are. Males are very easy because you know they've got a Y, so whatever they have, whether red or white, that is the genotype. So for example, here, we know we're gonna have Y. It's red. It's red. So that's a W plus. W plus is red, right? W is white. This is a female, no Y. White eyes. What does that mean? WW, because W is white. If you do the Punnett square, you'll see something. First of all, the only thing that the mom can pass down is W, right? She's WW. So the only thing she's going to pass down to either of the progeny here is W. The male can pass down an X, which in this case is a W plus, or a Y. And that's where you get the F1. See, red-eyed female, X and X. White-eyed male is a Y chromosome. Now, when you look at this, again, what is wrong? Well, half of them are red-eyed and half of them are white. There's nothing suspicious there necessarily. But each gender has only one color. Look, all females, red eyes. All males, white eyes. This wasn't making sense at all, right? This wasn't making sense at all. And so, and again, you can do your Punnett square. What you would get was W and W. And usually, again, in class, I'm asking all sorts of questions like, what type of fly is this? And then people would say white-eyed female. And then I can do W plus from here, W. And this was a red-eyed female. And now we can do the W again, but now we're switching to the next chromosome. This is a Y. And so that's going to be a white-eyed male. And then the fourth one on the Punnett square, W plus Y. So see, our Punnett squares still fly. Well, that's a bad way of saying it. They still work. Okay. Red. Yeah. And what they would see is they would get these in 1-to-1 ratios. And so again, this wasn't making sense. Why are all the male flies white-eyed? Why aren't some of them red? Same thing with the females, right? Oops. Okay. Well, we had microscopes. We had microscopes at this point. And so one of the things that they started to see was that the mutant phenotype, right, the YI was inherited in the same manner as the X chromosome. So they were looking at, remember colored bodies? We did this the first time we met, chromosomes, colored bodies. These colored bodies and the way that they were being passed on from one generation to the next actually matched the way the white color was being passed on from generation to generation. And so when they looked at that, they came up with another type of inheritance, which we're going to be doing. They came up with X-linked inheritance. So it can be autosomal, which means it's on chromosomes 1 through 22, or it can be X-linked. Now, there is such a thing as sex-linked, in which case it's the X and the Y. But again, the Y doesn't have very many genes on it, so it doesn't really play a big role. So some people say sex-linked, some people say X-linked. We use it interchangeably. Okay, I usually say X-linked because that's what we're studying usually in our examples. It's X-linked, but X-linked is sex-linked. Okay, so how did they resolve? How did they come to figure out precisely what was going on? I'm going to show you what proved that our chromosomes are what carried our genetic material. This now loops into the first lecture we've had in this course, which is, okay, but what on the chromosome was the genetic material, the DNA or the protein? But before that, they didn't even know where these determinants, the things that determine what we look like, were. And that's what we're about to see. Okay, this is a white-eyed female. So we all know what the genotype is. WW, because W is white and it's recessive. This is a red-eyed male, so we know exactly what that genotype looks like. There's a Y, because it's a male. And this must be W plus, because it's red. And so this is white and this is red. If you cross these, what you would get is W plus W, and this is going to be red female. Okay. And you're going to get plus, then you're going to get WY. And that's going to be a white male. But something happened. Something happened when they were doing these experiments. What they saw was 1 in 2,000 flies were either a red-eyed male or a white-eyed female. Okay, let's everybody look at the flies. Everybody look at the flies, right? WW, W plus Y. If you do the Punnett square, there is only 2 things you can get from this. W plus with a Y. I'm sorry, W plus with a W, or a Y with a W. And here they are here. Can you get a red-eyed male? The answer is no, you can't. Can you get a white-eyed female? No, you can't. So where in the world did these flies come from? Because there they are in the bottle, they exist. How in the world could that be explained? The way they explained it was something called non-disjunction. Let's look at the left. We know that when we're making sperm and eggs, right, we undergo 2 rounds of meiosis. So in the first round of meiosis, we go from 4 copies to 2, and in the second round of meiosis, we go from 2 copies to 1. And you get normal separation of chromosomes, right? Mendel even talked about this, equal segregation. And these are gametes. These will either be sperm or they'll be egg. What non-disjunction is when by accident, these chromosomes do not separate. And what happens is you have one egg that has 2 copies, or one sperm that has 2 copies, and another egg or sperm that has none. Now, usually this is not survivable in humans, right? If this happens with chromosome 1, not survivable. If it happens with chromosome 2, not survivable. There is only one chromosome in which this is survivable. We're going to talk about that a little bit later. Okay. The answer to that is 21. This is what they found to happen in the fly. Remember, these flies are impossible. They're looking at combinations that could not exist. This is what they saw. So in the male, again, here's your male, and the sperm. And what they found was in the egg, you had non-disjunction. It doesn't necessarily have to be in the egg. The non-disjunction could be in the sperm as well in any particular case. But in this case, because of the alleles, see the alleles in the sperm don't match the alleles in the egg, they're totally different. They were able to see what happened. And so what is it that you get here? W, W, W plus. What do you get here? W, W, Y. What do you get here? W plus. What do you get here? You get Y. Now, the genetics of flies are a little bit different. W, W, W plus, turns out that combination of chromosomes is lethal. They can't survive that. A fly which has just Y, well, that is lethal. It won't survive that. If you have a fly and you get WWY, that turns out to be a white-eyed female. And if you get a fly that is W plus, that turns out to be a red-eyed male. As it turns out, and what we know today, in flies, what controls sex is the number of X chromosomes. That's different than in humans. What controls sex in humans is the presence of the Y. So it's different. In flies, if you have 2 X's, you are female. If you have 1 X, you are male. You are male. And that's different from what we see in humans. And so what they could do is now is because we had microscopes, they could look at the cells of the flies and realize that the flies had an abnormal number of chromosomes, and they could follow the chromosomes from generation to generation, and then they could follow the eye color, the red or the white, and these impossible flies that existed, right? What you're seeing there on the screen, these flies that were impossible would be, actually these aren't, because this was supposed to be a red-eyed. Anyway, that's okay. No, this is right. Yeah, the white-eyed female, that's the one that was impossible. The red-eyed male, that's the one that was impossible. So these guys were impossible, but there they were. And when they took cells from these flies and they looked underneath the microscope, they noticed that the sex chromosomes were abnormal. And this is what proved, because remember, these flies were supposed to be impossible, but then you look inside the cells, you're like, these combinations of sex chromosomes shouldn't look like this because there's 3 of them, or 1, but not 2. This is what finally proved that it was the chromosomes that carried our genetic information. Okay, and down the road, as we all know, people start to fight about, okay, well, what on the chromosomes carry the information? Because they were made up of protein, DNA, and RNA. Okay, and so this is when we finally knew that those little colored bodies was carrying all of our traits, right? And so again, in flies, sex is determined by the number of X chromosomes, not by the Y, right? So in females, you're gonna have 2 X's, and females and males, you're gonna have 1 X. If you have 3 or 0 X's, it turns out to be lethal. It turns out to be lethal. Okay, what we're going to do now is work down the home stretch. It looks like we're going to finish very early, actually. We might finish an hour early. So let's go ahead and take a break. Let's take a 10-minute break. And we're going to come back. We're going to do the last part. We're going to be down the home stretch. And then again, I'm going to finish off midterm 2, and then I'm going to send out an email with regard to the quizzes, which will be open in the last 24 hours leading up to the final. And again, if that didn't make sense, then you can wait for the email. Okay, so let's go ahead and take a break. Let's come back at 3:20, and then we will be down the home stretch. Okay, let's take a break. Okay, what we're going to do now is we're going to switch gears like we did last time, and go to humans. So before we did pea plants, and then we went to humans, and now we're going to do the same thing. We started with flies to see how it all started and how sex chromosomes were discovered and how they worked, and now we're going to switch to humans. Okay, as I said before, with humans it's a little bit different with regard to the determinants of sex. With regard to biological sex, it really is the presence of the Y chromosome that is going to trigger certain things. And so when we think about biological sex in humans, the default pathway is female. We all start out with the default pathway. That is, we all start out with the pathway of being female. So what is it that changes in approximately half of the individuals? That has to do with the signal that comes from the SRY gene, which we see here as the sex determining region of the Y chromosome. And we're going to see how this works, okay? Particularly because we're going to see how the errors in some of this mechanism had led to our understanding of how we are triggered with regard to biological sex. And so, you know, it's interesting. I get a lot of these, you know, with regard to sex determination, I get a lot of questions, you know, in terms of like hermaphrodites. I get questions with regard to people who whose chromosomes are a little bit abnormal. You know, can you have an individual who's XY but female? Can you have like quadruple X? And it's in human genetics that I go through all of that. But usually the answer to all those questions is yes, which is very strange. But the question is, how does that work? Let's take a look here on the screen. On the left side, we're going to see the way we activate a normal male. Okay, and in the absence of what we're going to see, then we go the default pathway. Remember, our default pathway is we all start off as going towards female. Let's take a look here. Here's the Y chromosome here. Okay, here's the SRY. That's that testis determining factor. That's that region of the Y chromosome that determines sex, right? So what's going to happen is these genes are going to then make the secondary organs. In this case, you're going to develop testes, and those testes are going to then start to produce testosterone. This testosterone is that signal that's then going to cause all of the other tissues that are sex-related to start going male. And so if this gene gives rise to tissue which produces the signal, then where is the signal going? This signal is going to a lot of the tissues that can go one of two directions depending on male or female. But the important thing is that those tissues have testosterone receptor. In other words, I can give a cell anything. I can give it testosterone, I can give it estrogen, I can give it interleukin-2, I can give it interferon gamma, I can give it all sorts of growth factors. But that cell may not care what I give it. The question is, does it have the receptor? It's like a lock and key. I can give you a key, right, to an office. And as you're walking down the hallway, there might be 24 doors. Now, that doesn't mean just because I gave you a key, you can just walk into any of the doors. Your key must match the doorknob. And if it does, you have that lock and key mechanism, in which case you activate the lock and you can open the door. Same thing in our biology. There are lots of hormones, lots of growth factors that are secreted throughout our body, but those growth factors, those hormones, they don't activate every cell in the body. For any cell to get activated, it must have the receptor, which means that the doorknob fits the key. In this case, the testosterone is the key. And the question is, do the cells have the doorknob? Well, that gene that creates the testosterone receptor is on the X chromosome. So what is happening here? The gene on the X chromosome is being expressed, and now we have testosterone receptor. Remember, regardless of who you are, you have the testosterone receptor gene activated, and it's on the surface of your cells. And as you are developing, right, during embryogenesis, different tissues of yours will express the testosterone receptor. And the question is, will those cells ever see testosterone? If they don't, then those tissues continue to develop female. But if there's a Y chromosome, then this signal, which is testosterone, is given off, and all of those tissues now get activated because they are displaying the testosterone receptor. And now you start to get all these male secondary sexual characteristics because the tissue starts developing male. That's the signal, right? That's the signal. So the signal comes from the Y, but the receptor, which we all expressed during development, comes from the X, because every one of us has the X. Now, something happens in some individuals, which gives us somewhat of a strange situation. Let's take a look. Here we have an individual that is XY, an XY. Here's the SRY gene, right? That is causing the development of testes, and the testes produce the signal. But what do we see here in this individual? The testosterone receptor gene is mutated. They were born with a defective receptor. Their doorknob doesn't work, even though keys are being let out everywhere. Because there's no testosterone receptor, the signal is being given, but it's not being received. What does this mean? It means that those secondary traits continue female, while some tissue is male. So people ask me, is it possible to be XY female? And some people laugh, oh, of course not, it's not possible. I mean, there's a Y there. But actually it is. We do have XY females. And it is because the testosterone receptor on all of their tissues doesn't work. And it's a gradient. It just depends on the mutation. Some mutation is so severe that you don't get any reaction with testosterone on that receptor. So everything looks female with regard to the secondary sex tissues. So much so that some females don't know their genetic makeup until they try to have children. When they try to have children, there's infertility, they go to the doctor, a bunch of tests are done, and it comes to find out that they are XY. And XY females are sterile. Okay. But other mutations, and there's dozens of different types of mutations on the testosterone receptor that we've identified. Some mutations are not total mutations. Sometimes the receptor works a little bit, and that is worse. You would rather the receptor not work at all. Because if it works a little bit, then what happens is some of the tissue does start to change. And it does start going somewhat male, but not all the tissue goes male. Some of it stays. Why? Again, because you have a small level of effectiveness. And so that produces now characteristics where some individuals have traits of both genders. And sometimes you can see it, sometimes in facial features, you're not really sure what's going on, body shapes, the development of breast tissue. Obviously, anything in the groin area is hidden, so we don't see that. But there are other areas of the body where we do see, to some extent, and you're sometimes caught wondering because they have both. Okay, and so we do have XY females, and it is on a spectrum because we've found so many different mutations. When it is an absolute mutation, and that protein, which is the testosterone receptor, when that thing does not work at all, you tend to get people who look kind of normal, but they can't reproduce. If the receptor does work to some extent, you start to get some of the signal received. And so you start to get some maleness, but in some areas you don't get the signal received, so you get some femaleness. And so again, in human genetics, we go more into this, but this is the determinant in humans. We all start off female. That is our default for every one of us. Our default is female. And then if the signal is given and received, then you go down a different type of differentiation in those cells. So the tissues develop differently, okay? If you don't have that, it's called androgen insensitivity syndrome. Androgen is just a fancy word for male hormone. That's it. Androgen is just a fancy word for male hormone. So what this is saying is you're insensitive to this. In other words, the signal was given, but you're insensitive. You don't react to it at all. Why? Because your testosterone receptor doesn't work. Okay, and it can be partial or complete, and you would want complete, because at least physiologically, even though you may not be able to reproduce, physiologically, you look what in society we would consider normal, right? Partial, you're going to start to get both. Okay, and that's a little bit more traumatizing socially. Now, there is another way. Actually, I'm sorry, let me restate that. There are other forms of defects with regard to these sex hormones. We saw that in Drosophila, you can have XXY and X null. We call this X null, right? Well, in this case, it's a fertile female because you have both X's. In this case, it's a sterile male. You're missing one of the X. You're missing the Y. In humans, you can also have this. We have humans that have XXY and X null. It's survivable. In humans, if you have XXY, because it's got the Y, right? It's got the Y, then this is actually going to be a male. This is going to be a male. And in this case, what happens is, because you have overexpression of the genes that come from the X chromosomes, there's an imbalance in a lot of the proteins that are being expressed in our cells, in the entirety of the physiology, not just in sex organs or secondary sex tissue, but in all the tissue. And so what you see in these males is they tend to be tall, and they tend to be very very skinny. So they're very gaunt, right? They have this height, they're very skinny, and there's usually mental deficiency there. We used to call that mental retardation, where the term retardation meant that there was a slowness or a deficiency, right? We've gotten away from that word, but it's this mental deficiency where you cannot really rationalize or function that normally. And they are also sterile, okay? And so these are Klinefelter males, When you look at the X null, because there is no Y, this turns out to be female. But again, you have an underexpression of X, right? Over here in these males, you had an overexpression of X. We're only supposed to be expressing off of one. Females are supposed to be expressing off of two. That's the balance, that's the homeostasis. And so in this case, what you're having is an underexpression of the genes that are all on that Y chromosome. And the Y chromosome is a pretty big chromosome, okay? So when you think about it, when you think about the size of the X chromosome, so remember, chromosome number 1 is the largest one. That's why it's called number 1. So you have chromosome 1 through 22, chromosome 1 being the largest, okay? And then chromosome 22 being the smallest. And then you have an X and Y. The X chromosome is about the size of chromosome 7. So if you're going from 1 to 22 and you're at 7, that's a big chromosome. So chromosome X, there's a lot of genes there and they're not being expressed. Remember, this individual is supposed to have 2 X's. They're not being expressed to the point that they're supposed to. So this is since there's no Y, there's null, this is going to be a female, but in this case, the underexpression causes Turner syndrome. So what is Turner syndrome? It's kind of odd. It's almost the opposite of the male's Klinefelter syndrome, where in Klinefelter syndrome, males tend to be tall and thin, very gout. Well, in females, they tend to be very short and very stout. And they also have mental deficiency. And they're so stout, and that actually many people see it almost like a web neck. They say that, and I don't always see it when I've seen pictures of females with Turner syndrome, but in some of them you do see it. It looks like a web neck where it goes kind of from the jaw down straight to the mid shoulder and so forth. That's how stout they are. And again, there's the inability to produce, and there's that mental deficiency. So this is survivable, but obviously with a lot of physiological and mental problems. Okay. Okay, so let's talk about the non-disjunction that happens in human gamete. Well, yes, human gamete formation, either the sperm or the egg, and let's see how we actually get some of these syndromes. Okay, and we can see them on the screen. Now, the non-disjunction, if you don't remember, the non-disjunction is when we're producing egg and sperm, but the number of chromosomes is wrong because they did not separate. Remember what Mendel said? Mendel said that we would actually have equal segregation, that each one of the sperm or the egg being produced would get one copy of the chromosome. So what we're going to see here on the screen is how we actually get some of this non-disjunction then occurring and giving us these different syndromes, right? Okay, let's take a look. So on the left-hand side, what we're going to do is we're going to see where we have the development of eggs. So see up here up top, we have an X and an X. And what we're going to do is we're going to do our final meiosis 2. In our meiosis 2, remember, these are supposed to separate. So you get an X in one egg and an X in another, but non-disjunction occurred. What happens? This egg gets nothing. This egg gets 2. Now, in each scenario, what we're going to do is we're going to see its fertilization. So here you have a sperm with an X and a sperm with a Y. Same thing over here, a sperm with an X and a sperm with a Y. Okay, and these are normal. And so what you can get here is you're going to get this X null. There's our Turner syndrome. This is survivable. This is survivable. This female does not need anything on that X. This is Y null. Well, all of us, regardless of what biological sex we are, we all have an X. You will have 1 or 2. So because this individual has no X, it's not survivable. Let's go over here on this side. Here, the non-disjunction produced 2 sex chromosomes, an X and an X in that egg. And so over here, we have an XXY. That's our male. That's our male Klinefelter syndrome, right? We can survive the 2 X's with that Y, but it causes all those problems, right? Here we have an XXX. We have a triple X. This is actually survivable too. This is actually survivable too. We found, my knowledge is we have found females with up to 5 X's, 5 X's. Okay, and so obviously that's a big mess. We've seen 4. Here what we're seeing is 3. And so what we have here is, again, a lot of developmental problems because we have more overexpression of all of these genes. Okay, let's look over here. In this case, the egg has the appropriate separation. So the eggs are down here, right? The eggs have the appropriate separation. So in this case, each egg has an X, right? The eggs can't have Ys. And now what we have is where the gametogenesis is producing sperm. In this case, the sperm here, we have non-disjunction. The sperm is null. It doesn't have anything. Well, I shouldn't say that. It doesn't have a sex chromosome. The other 22 may be just fine, but the 23rd, which is the sex chromosome, it did not receive. The non-disjunction made it so that the X and the Y are over here on this side in this sperm. What happens? You have normal eggs. Here you have a fusion. You have X null. Here you have your female Turner syndrome again. And over here, where you put them together, that's your XXY. Now you have your Klinefelter male again. Okay, so there's different ways in which you can develop these defects. And many times, there are people within the population that we wouldn't suspect have these chromosomal abnormalities. It just depends. With Klinefelter and Turner syndrome, it's usually pretty obvious, but there's other defects which we're not going to go into, which is on a spectrum. And so sometimes you do have people who are not what you would typically expect genetically, but who function well enough that you would never suspect. And sometimes even they don't know. Like I said, sometimes it's not until that they try to procreate that they go and get themselves checked and they're told there's some abnormality here. And so when we look at the X and the Y, obviously the X is much bigger. It carries a lot more genes. They're not necessarily sex associated genes because we all have them. So there is some physiology that has its genes on the X, but has nothing to do with sex itself. The Y chromosome, very small. And so when we think about the regions of the X chromosome that have genes, females can be homozygous and females can be heterozygous. But when we think about males, they cannot be heterozygous or homozygous. When it comes to the genes on the X chromosome and even the Y chromosome, males are said to be hemizygous. Remember, hemi means half, right? Like hemisphere. So males are hemizygous with regard to genes on the X and the Y chromosomes. Females can be heterozygous or homozygous with regard to genes on the X chromosome. We've gone through this already, so I'm going to kind of skip this. The Y chromosome, we're still studying it. Not a lot of genes there from what we can tell. Okay, I'm just curious because I always ask this in class, and this is a small class. Usually my genetics classes are like 60, 90, and I always ask this question. Is there anyone in the class that is red-green colorblind? So I know we're a small class. Anybody here red-green colorblind? You can put it in the chat or unmute. No? Okay, so I usually ask, and when my classes are big, usually every other time, like every other class, I'll have somebody who's red-green colorblind. One time I even had two. Now, the receptors for color that reside in our retina, there's 3 of them. You've probably heard of the rods and the cones and all that in your previous biology classes. But what happens is the ones that detect red and green are on the same chromosome, and it happens to be the X chromosome. What does that mean? It means that females have 2 copies of these receptors, but males only have 1. So if a female has a mutation in the receptor, there's always the other X chromosome. That has copies that probably work. So you will rarely, even though it's possible and they exist, it is rare to find a female that is red-green colorblind because they would have had to inherit that mutation from both mom and dad. And if it's a consanguineous mating, remember, consanguineous means people are related. If you have cousins marrying, that's a little bit more feasible. But in the general population, that'd be very rare. In males, what we see is males have 1 copy. So if you get the mutation, you're out of luck. There is no second copy to rescue you from that. So whenever I've had people, and I've had many over the years, again, it seems like every other time I offer genetics and it's a large class, I ask, every other time I'll have at least 1, and it has always been male because you only need 1 mutation. The other one's just a Y chromosome. It's not doing anything with regard to sight. And during class, if I do have someone that is red, green, colorblind, I'll ask them, I say, do you see all the dots on the screen? And they'll say, yes, I see the dots on the screen. I said, well, there are some dots that are a different color, and they form a number in the middle of this big circle. I said, can you see what number that is? And they'll say, no. And then I asked the rest of the class, I said, what number do you see? And they'll say 16. And you can always have that jerk in the back goes 22, but no, it's 16, right? And you can probably all see it. The green circles are showing us 16, and they can't see that, okay? Which is why when you think about traffic lights, which use the exact same colors that are on this slide, right? Number one, the green on traffic lights, when they make them, the spectrum of the color that's given off by the green bleeds into the blue field so that red, green, colorblind can see it better. It bleeds into the blue. Sometimes I see it's almost aqua. That is not even green. But even the ones that look green, it bleeds into the blue field so they can know. The other thing is that, and you may not know this, but I'll ask you, and again, this is kind of some of the fun that we have when we're face-to-face, these types of conversations, but I'll ask you, which color on a traffic light, which of the three colors is the top light? And of course, half the people are saying green, half the people are saying red. 90% of them don't know, they're just guessing. And so if you're red, green, colorblind, the other thing that you know is that red is the top color on a street light. And so depending on what is lit, now that may become tricky at night because from far away, you're not sure which circle that is. Is that at the top? During the day, it's much easier because you can see the thing from far away because the backdrop of the sky is blue. But at night, that can be a little bit tricky. You know, I can see it's lit, but is that the top one? Is that the middle one? Is that the bottom one? So again, they make it so that it bleeds into the blue so they can have some, so they can detect some difference, okay? And again, X-linked traits, mostly you'll see in males, not females. Okay, so let's do this problem. Let's do this problem. If a normal man marries a woman, we're looking down here, right? Looking down here. If a normal man marries a woman with normal vision, whose grandfather was red, green, colorblind, what will the probability of having a colorblind son be? And so this is what we're going to do. We're going to use a different nomenclature. CB is colorblind. CB plus is normal. We're going to use this nomenclature so that we continue to get better at this nomenclature, okay? And so sometimes what will happen in textbooks, what you'll see is, for example, you'll see something like this, CB / CB. All this is saying is this CB on this side is on one chromosome. This CB on this side is on another chromosome. Well, what's the point? And that's because if you drew it like this, see CB, CB, if you drew it like this, some people might confuse that for two genes because we've been doing that, remember? So some people might say, okay, well, this is B, B, so there's one gene, small b, small c, small c, and they might think these two are the same. CB, CB is the same as small b, little c. So we don't do it like this, right? So sometimes when you start to look at the textbooks, they start to separate it here. So you know, okay, CB is on one, and this CB is on the other, and that's one gene. So we don't do it like this. Okay, let's give it a try. We're still practicing now, right? Now remember, this is X-linked. This is X-linked. And we're doing a different nomenclature. So we're ramping it up here. First things first. This is recessive. Fill in the affected people first. And with males, again, in an X-linked trait, it's super easy. Let's take a look. Colorblind. CBY. Colorblind. CBY. Okay. Now remember, for an X-linked trait, men are very easy because they only have one copy. So in this case, normal, CB plus Y. Normal, CB plus Y. Normal, CB plus Y. Any other males? No, that's it. Females tend to be a little bit trickier, again, because we have two copies, so we kind of have to figure out which one is the recessive one, which one we can't see. But that's doable. Let's take a look. Okay, this female, normal, so CB plus, and see, she's marrying in, but we do see kids expressing the disease. So let's take a look. In this case, let's do the mom here. Let's do the mom of this individual. The Y, see here, the Y came from dad. So where did this CB come from? It came from the mom. The mom is normal. The mom is not colorblind. So the mom is CB plus. CB plus CB. There's the colorblind trait. Now let's go back up. Well, this CB here, this CB came from dad, right? This CB plus came from mom. So this disease allele is not from mom. This is a rare trait. Since we know it didn't come from mom, mom is probably CB plus. Again, we figured out that the reason she has a grandchild that's colorblind is because of him. So chances are it's not because of her, because colorblindness is a rare trait. Okay. Okay, let's look over here. So dad passed down the CB, couldn't pass down the Y because she is female. So this would be CB plus. That's from dad. What did mom pass down? Well, mom can pass, and you can do the Punnett square on this, but mom passed down, we don't know. But because you're a genetic counselor, what is your concern? You want to follow the diseased allele. And what's the chances of that? 1/2. Either CB plus or CB, we don't know. So that's 1/2. Thing is, this female is the same. 1/2 CB plus CB. Okay, now we got a Punnett square over here to make. So here we're going to do dad. So dad here is going to be CB plus Y. And then over here, we have mom, and mom has CB plus and CB. Okay, remember CB plus is normal. And now we can fill this in. So this is CB plus CB plus. This is CB plus Y. This is CB plus CB. And this is CB Y. Okay, what's the question now? What will the probability of having a colorblind son be? Well, we look over here. This child can be born any one of these four. But the probability of being a colorblind son, 1/4. And it's this one, the one on the bottom right, CB1. The probability of being a colorblind son is 1/4 from here. Okay, but remember, we're not done. We got to take into account everybody who contributes to the genetics of the child. So we go up the left-hand side, 1. We go up the right-hand side, 1/2 times 1 times 1 times 1 times 1. Okay, now we take the child here into account, 1/4. One-eighth. 1/8. Okay. I have another question. I'm going to change up this question at the end. So the question is, what will the probability of having a colorblind daughter be? So I'm going to wait for answers in the chat. What's the probability of having a colorblind daughter? I'm talking about this diamond here. What's the probability of having a colorblind daughter? Good. Jessica and Julian and Gwen and Elizabeth, very good. When you look at the Punnett square, or when you look at the Punnett square for the parent, these are these two parents, and you look at the child. Okay, these are male. These two are male, so that won't work. So you're left with 2. In this case, both of these are normal. In this case, one of them is normal, but it's dominant. So you're right, it's 0. Very good. What will be the probability of having a colorblind son? 1/8. Colorblind daughter? 0. 0. Okay. One thing that didn't pop up here that I want to kind of show. Remember in previous examples, we had little a, and you're like, well, this person's unaffected. So this person cannot be little a. So it's 2/3. It's 2/3. One of the things you want to do is you want to be careful, because when you do these Punnett squares, sometimes you eliminate people based on gender. So for example, let's do these two individuals here. Let's do these individuals here. I'm going to do CB, and this is no longer part of the problem on the screen. I'm just kind of wanting to make sure I make a point that you guys see. And then she is CB plus Y. No, sorry, she's CB plus CB. Okay, now what I'm going to do is I'm going to fill these in, CB plus, CB plus, and this is CB plus Y. This is CB plus, sorry for the chicken scratching, CB, And now this is CB Y. Okay. See this, CB plus Y, CB plus CB. And I asked, what are the chances that she is a carrier? She's CB plus. What are the chances that she's a carrier? In this case, and it wasn't obvious at the moment, she cannot be this. If she was, she would have a Y and she would be male. She could not be this. If she was, she would have a Y and she would be male. So this 1/2 is not 2/4, which is what some of you may have thought. Okay, yeah, 2/4. It's actually 1/2, 1 out of 2, like, but truly 1/2, 1 out of 2. Because there was 2 possibilities, and one of those is where she could be a carrier right here. So in this case, it didn't make a difference, but that's why I always say, I always write out the Punnett square. It may be that you have to eliminate some based on affected or unaffected, right? If it's an autosomal, big A, little a, and you know, in a situation like this, we could have done the big A, like, but in some cases it doesn't work. In this case, it would have worked fine. We could have done something like this. But the point that I'm trying to make is always be aware that you might have to eliminate certain ones. And it might be maybe just one of the 4, not 2 of the 4, where you're like, you know what? This person cannot be this. So you eliminate it. And now you're, in this case, 1/2. In some cases, we've had 2/3. That's been common, right? But always when you're doing the fetus, right? When you're doing the embryo, we don't eliminate because we don't know what this person is. So all 4 are fair game. It's only when people are born and we know this person's affected, this person's male, this person's unaffected, this person's female. These are things that we know. So we might have to cross those out. Okay, so be careful. I always, it doesn't matter how long I've been doing that. I've been doing this 20 years. I still draw out my Punnett squares. Okay, it takes 5 seconds to do one. Okay, we're gonna do a little exercise here. Using the same nomenclature as before, I'm going to ask all of you to, well, if you don't have a paper or pen left, I do this during class. I ask everybody to fill in this family pedigree with CB, CB plus, and Ys, the way that we did. So if you want to give it a shot, I'm going to give you maybe a 3-minute head start, and then I'm going to start to fill it in. Okay, so I'm going to give you a 3-minute head start. If you want to draw it out on a piece of paper or do a screenshot and use another program to type it in, that's fine. So in 3 minutes, I'm going to start filling stuff in. Okay, go. Let me know if you have a question on any particular person. We've got 4 generations and all the people are numbered. Okay, I'm going to start filling in. Okay, I think that's all of them. Are there any questions to any of the individuals on the screen? No? Okay, great. And so I have a kind of question for everybody. We're down the home stretch. We're about to end. So genetically normal females are XX, right? And genetically normal men are XY. So the question is here, do XX females have twice the amount of gene products, in other words, a protein, right? Do XX females have twice the amount of protein encoded by the X chromosome as males? And initially you would think that, well, yes, because they have two. But it turns out that that's actually not the case. One of the things that happens in females is, and we can see this here, sometime at around the 100 to 200 cell stage, when females are developing, right? At this stage, anywhere between when the embryo is between 100 and 200 cells, each one of these cells, each one will decide to turn off an X. It might be over here. So we have the paternal X, we have the maternal X. And in this case, you have the maternal X inactivated. Over here, we have the paternal X inactivated. And so what happens is these chromosomes that are inactivated, they get methylated everywhere. So they get extremely inactivated. From then on, let's take this example here of this cell here. From here on, every cell, remember we're 50 to 70 trillion cells as a human, every cell that comes from this particular cell, see this one here? Every cell that comes from this particular cell will always have that chromosome inactivated. So in this case, this cell turned off the maternal, right, and kept only the paternal. Or did I get that backwards? It doesn't matter. The point is this cell at the 100 to 200 cell stage during your development, if you're a biological woman, from then on, that cell, which went on to make many, many, many hundreds of billions or trillions of cells, the same X is inactivated. And so it is in each cell that you don't have twice the expression. You don't have twice the expression. What happens though is in the other inactivated X, there is some leaking. You do get some about 10% expressed from the inactivated X. That's important. That's why even if you have a mutation, as a biological woman, one of your X's is mutated in a gene, the other one, even though it is inactivated, there's still some leakage. There's still some protein activation to the tune of 10 to 15%, which is why you can overcome that mutation. Men don't have that, right? Men don't have that. So if you've mutated, that's pretty much it. Okay. We see this inactivation better in cats because cats have fur and the fur is on the X chromosome. So in this case, you see that here we have an orange and here we have a black. These are the pigments on the two chromosomes, on the two X. Here we have X inactivation of the black, which means that fur is orange right there. Here we have inactivation of the orange, which means the cells that come from this cell for the rest of that cat's life are going to be black. So we see it more so in cats because they have fur than in human females because there's no physical marker. And the last thing is, are there any male calico cats? So in this case, you have orange being expressed on one area of the cat. You have black being expressed on another. But that's because you have two X's, right? This came from one X. This came from the other X. Are there any male calico cats? Well, the answer to that is yes. How is that possible? Okay, well, number one, they're very rare. Number two, they're very expensive. Okay. There are about one in about every 3,000 calico cats is male. But how does that happen? That's how it happens. There's a non-disjunction. This Y makes the cat male, but the X's give it the pattern. 1 in 3,000 calico cats. I hear they're pricey. I'm not sure what pricey means. And so the rest of these are this is a summary slide, and that's it for sex linkage.